Good afternoon. I'm Dr. Brian Shapiro, a marketing segment manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled, Get to the Heart of Your Toxicological Studies with Immortalized Primary Cells, and it's presented by Mr. Jonathan Sehgal and Dr. Sujoy Lahiri. Mr. Sehgal is a product manager, while Dr. Lahiri is a lead scientist in R&D at ATCC. In this presentation, our experts discuss the characteristics and utility of some of the various renal and cardiovascular models in toxicological assays. Mr. Sehgal will start by sharing ATCC's mission and future direction and then delve into our immortalized primary cell portfolio. Dr. Lahiri will then show some compelling data from case studies that feature both our primary immortalized kidney and cardiovascular models. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome our first speaker, Jonathan Sehgal.
Thank you, Brian, and good morning to our audience. I'd like to spend a few moments to introduce you to our organization. Founded in 1925, ATCC is a non-profit organization headquartered in Manassas, Virginia. ATCC curates and distributes the world's largest and most diverse collection of biological material. This resource is the gold standard for cell culture-based research. In addition to maintaining our core collection, ATCC has an innovative R&D center in Gaithersburg, Maryland, where we develop advanced complex in vitro cellular models, such as the ones we will be discussing today. At ATCC, we think about advanced in vitro cellular models as the next stage along the evolution of cell culture systems. It all started with the continuous cell lines, such as HeLa, A549, or CACO2, which were isolated from tumors. The next generation of models included engineered cell lines, which were modified via transfection or transduction to include a reporter signal. Along with these models came the development of isolation procedures and media to support the in vitro culture of primary cells isolated directly from normal or diseased tissue. The third generation of cell models included CRISPR-edited cell lines, the discovery and establishment of induced pluripotent stem cells, and the development of immortalized primary cells, which will be the focus of our webinar today. Over the past decade, the fourth generation of cell models has focused on three-dimensional organotypic culture, such as spheroids and organoids, which give researchers the possibility to culture cells from tissues that are otherwise difficult to maintain in vitro, such as intestinal epithelial cells. More recently, microphysiological systems, such as organ-on-a-chip approaches, have been pioneered to build models with more complexity and physiological relevance. Depending on their research objectives and cell model availability, scientists can and oftentimes will utilize models from across these different generations. ATCC, with its broad collection of in vitro models, from authenticated first-generation cell lines through to high-quality inputs for the construction of a fifth-generation microphysiological system, is here to support all your research needs. Cells are one of the most important inputs for predictive in vitro models, and there are many aspects to consider when selecting the cell source for your research. Now I'll give more details on advantages and challenges associated with each of these cell sources. Broadly, we classify them into four different types, the first of which are the cell lines. These are mostly derived from tumors and are highly proliferative, easy to culture in vitro, maintain a stable phenotype, and can be fluorescently labeled. However, working with cell lines has some limitations. Often the in vivo functions of normal tissues may be compromised, and the cells may differ genetically and phenotypically from a healthy tissue. The second major source of cells are primary cells. In most cases, these are the most representative model of healthy in vivo cells. Primary cells maintain many of the important markers targets, and functions of healthy tissues, resulting in a model that can give high predictability of physiological behavior and response. Working with primary cells does, however, present some challenges that need to be taken under consideration. These cells have a finite lifespan in vitro, and in order to continuously work with primary cells, multiple donor tissues will be required. Cells from different donors may result in lot-to-lot variability, which can be advantageous as this represents the normal human diversity. However, if a stable phenotype is required across studies, researchers will need to screen multiple donors to identify suitable cells. This can add time and cost to a research project. The third major source of cells are those derived from induced pluripotent stem cells, or iPSCs. These offer some unique opportunities to generate cells from multiple tissues with the same genetic background. iPSCs also have extensive proliferative capacity and can be used to generate large banks of uniform differentiated cells, overcoming the need to source new donors during a program. These cells also have a high therapeutic potential, with several ongoing clinical studies involving the use of iPSC-derived cells. Limitations when working with iPSCs include their complex culture requirements, extended timelines required to generate differentiated cells, and the poor maturation of these differentiated cells. Many of these models have been only able to show a fetal phenotype. The third cell type we'd like to mention are immortalized primary cells. These cells have the dual advantage in that they maintain similar genetic and phenotypic features of the primary tissue of origin, along with a similar functionality to the primary cells. However, due to the immortalization process, they proliferate in vitro for longer than primary cells, enabling scientists to access a cell model that both represents normal human physiology and maintains a stable phenotype over time. Let's take a few minutes to talk about the technologies used to immortalize primary cells. In normal cells, mitosis is a tightly controlled process to ensure cells do not continuously proliferate, which could result in tumor formation. Cell immortalization methods look to interrupt one or several of these mechanisms to enable indefinite proliferation. Enhanced telomerase activity, which extends the telomeres during each cycle of cell division, has been identified in cancer cells. This enhanced activity ultimately plays a role in the replicative immortality of the tumor. Primary cell immortalization takes advantage of this process by artificially activating the usually silent telomerase in somatic cells. Some other methods for immortalization include transfecting primary cells with human oncoviruses, such as simian virus 40, Epstein-Barr virus, or human papillomavirus, which contribute to the activation of the telomerase gene. Finally, intervention with the expression of many of the cell cycle regulatory proteins, or withdrawal of alternative tumor suppression mechanisms, all can lead to cellular immortalization. At ATCC, we have expertise with multiple methods of cellular immortalization and utilize many of the strategies described. Our most widely used method of immortalization involves the stable expression of human telomerase reverse transcriptase, or the hTERT protein. For some cell types, such as our recently launched HPAEC BMI1 cells, we have found that using alternative cell cycle control genes, such as BMI1, are required to achieve cellular immortalization. I'd like to provide some more details around the use of hTERT to create immortalized primary cells. With every cell division cycle, the ends of the chromosomes, known as telomeres, are progressively shortened and eventually lost. Shortened telomeres will trigger cellular senescence and in turn cell death. Telomerase, which is active in germ cells, stem cells, and some cancer cells, overcomes this limitation. As seen in the figure on the right, the hTERT reverse transcriptase ribonucleoprotein complex adds tandem repeats to the end of the linear DNA. This rebuilds the telomeres and protects the integrity of the chromosomes, allowing cells to continually proliferate indefinitely and escape senescence. As mentioned, ATCC has extensive experience using hTERT as a method for immortalizing primary cells. On the left of this slide is the process of generating our hTERT immortalized primary cells. Using a lentiviral transduction, we stably transfect the plasmid containing the catalytic subunit of hTERT into the primary cell of choice. After antibiotic selection, we perform extensive characterization of a single cell colony, which we then expand and bank. So let me tell you a bit more about the extensive characterization we perform with our immortalized primary cells. A few years back, ATCC released our hTERT neonatal dermal melanocytes. Take a look at the figure on the upper right of the slide, generated during the development of that line. This demonstrates the enhanced replicative capacity of an immortalized primary cell. The primary melanocytes are in purple, and the H-type immortalized melanocytes are in red. As you can see, the H-type immortalized population doubling rate is comparable to primary cells. However, the H-type cells continue to replicate, undergoing over 40 population doublings. For authentication, we confirm that the morphology and marker expression of the parental primary cell is maintained. In the panel with fluorescent images, you can see that immortalized primary melanocytes maintain a consistent expression of the melanocyte-specific marker TYRP1 and lack the expression of the fibroblast-specific marker TE7. Finally, we also verified the functional responses are within the expected range of a primary cell, ensuring the immortalized primary cells can be just as predictive and physiologically relevant as the parental cell. As an example, in the graph at the lower right corner, we demonstrate the capacity of immortalized melanocytes to be incorporated into a functional three-dimensional skin culture. We grew either primary dermal cells, here in the yellow, or their H-type equivalents, shown in purple, in three-dimensional organotypic skin cultures. We then treated these cultures with Triton X100 and observed a similar metabolic activity in both the primary and immortalized melanocytes. I'm going to pass the presentation on to my colleague, Sujal Lahiri, who will discuss some case studies using our immortalized primary cells, working with cardiovascular and kidney cell types.
Thank you, Jonathan. So using the immortalization technique that Jonathan just spoke about, ATCC has developed several immortalized cellular models. These have proven to be extremely useful tools that researchers can take advantage of to further their research. Today, we are going to talk about two of those models, the kidney and the cardiovascular cellular models. Let's start with the immortalized cells from the renal tissues. As many of the drug metabolites are excreted through the kidney, our kidney constantly get exposed to potential toxic substances, which can cause drug-induced toxicity. We all know that the nephrons are the functional units of a kidney. However, one interesting aspect of the renal function is that our kidneys do not need to use all of the nephrons at once to perform its filtration function. Unfortunately, this large functional reserve of the kidney is the reason why we don't detect nephrotoxicity at early stage. Not until about 60% of our nephrons have stopped functioning that the kidneys begin to fail. Hence, nephrotoxicity is often hard to detect during a clinical trial. However, many drugs, such as the COX-2 inhibitors, can cause acute declines in renal function and glomerular filtration rate. Besides the liver, the proximal tubules of nephrons is a major site where the non-filtered xenobiotics are metabolized and excreted. The proximal tubules express a number of drug or solute carrier transporters, such as the organic anion and cation transporters, OAT1 and OCT2, along with numerous phase 1 and phase 2 drug metabolizing enzymes. Back in 2013, ATCC launched a new HTART immortalized human renal proximal tubular epithelial cells, which we named RPTEC, or the TART1, as this was also our first HTART primary cell. We did extensive characterization of the RPTEC cells by comparing these immortalized cells to their primary counterparts. We showed that the HTART RPTEC cells exhibit uniform expression of E-cadherin epithelial marker and the CD13 renal marker, and also form 3D dome-like structures, which are characteristics of the primary proximal tubular epithelial cells. Additionally, these cells showed a stabilized transepithelial electrical resistance, or TAR, This is a measurement of electrical resistance across a monolayer of cells, indicating the integrity of the monolayer. So, from an in vitro point of view, the RPTEC cells are the best representation of the human renal proximal tubular epithelial cells currently available, while at the same time, you can pass them in the lab at least 50 passages. For all of these features, we decided to use RPTEC or the TAT1 cells as a platform to create solute carrier protein overexpressing cells. As you can see in the figure on the left from the paper of Lin et al., OAT1 and 3 and OCT2 are solute transporter proteins that are localized to the basal membrane of the renal epithelial cells, highlighted here in red. They transport a wide range of relatively small hydrophilic organic solutes from plasma into the cytoplasm of the proximal tubular cells. The solute is then pumped out into the renal filtrate by ABC transporters. Because the activity of these pumps is often the limiting factor for solute clearance from the kidney, they are recommended by both the FDA and the European Medicines Agency or EMA as targets for drug-drug interaction studies. While this model is generally well accepted, the challenge is due to the lack of whole cell models that express SLC proteins, which I will explain over the next couple of slides. After the molecular validation, we wanted to see if the overexpressed SLC proteins localized to the plasma membrane as anticipated. So we fixed and stained the OAT1, OCT2, and OAT3 overexpressing RPTEC cells with the appropriate antibodies. The fluorescent images on the right shows their cellular localization. We see that the OAT1 transporter cells in the top panels, the red fluorescent signal depicting the OAT1 is clearly localized to the cell membrane. In the second panel, the OCT2 protein labeled with green fluorescence is localized to the plasma membrane as well. Finally, in the third panel, you see that the OAT3 clearly localized to the cell membrane of the OAT3 overexpressing cells. The last panel belongs to the parental cells, which is stained for OAT3. However, no staining is apparent as expected. So we set out to functionally characterize the SLC transporter overexpressing RPTEC cells. For the transporter uptake assays, we used two membrane permeable fluorescent dyes, a negatively charged 6-carboxyfluorescein or 6CF, and a small positively charged dye called ethanamidium iodide or EMN1. The purple lines in the three graphs of the upper panel indicate the SLC expressing cells, while green represents the parental primary cells. Solute uptake activity of OAT1 and OAT3 expressing cells in the top left and middle panel was assessed using 6CF as a substrate. The solute uptake activity of OCT2 was assessed during EAM1 as a substrate, showing in the top right panel. As expected, uptake of the fluorescent dyes increased with increasing substrate concentration in solute transporter expressing cells, but not in parental RPTEC cells, indicating that the observed transport is due to solute carrier protein expression. We further tested the functionality of the OAT and OCT expressing cells by inhibiting the transport function of the SLC transporters. OAT1 and OAT3 expressing cells were exposed to increasing concentration of the known OAT inhibitor novobiacin. The 6-carboxyfluorescein concentration and uptake time were held constant at 3 micromolar and 20 minutes respectively. Similarly, OCT2 expressing cells were exposed to increasing concentration of the known OCT2 inhibitor cimetidine, while EAM1 concentration and uptake time were held constant at 5 micromolar and 20 minutes respectively. We observed dose-dependent inhibitory effect for all three solute transporters in these uptake inhibition assays. Together, these kinetic assays results indicates that we have substrate-specific SLC transport into these cells based on the overexpression of the individual solute carrier proteins. Now, of course, the goal of having these SLC transporter cells is to incorporate them into real drug studies. From the outset, we wanted to see if the RPTech TART1 cells could be useful for studying drug-drug interactions. As we know that in cancer patients, there are a host of factors related to the disease and therapy that drive the development of renal failure. Many cancer patients have comorbidities, and many common prescribed medications interact with chemotherapeutics and also block the renal uptake of candidate drugs through the SLC transporters. We were able to set up a collaboration with Dr. Alice Gibson at the College of Pharmacy at Ohio State University. In order to study the drug-drug interaction application, Dr. Gibson and her then student Kevin Huang performed uptake assays using our RPTech cells, where they incubated some candidate drugs with radio-labeled known SLC substrate drugs in RPTech culture and monitored the uptake. If the uptake of the radio-labeled compounds were altered, then drug-drug interaction would be indicated. To test for possible drug-drug interactions, OCT2 uptake assays were performed with two radio-labeled drugs, the antibiotic levofloxacin or the anti-diabetic medication metformin. These uptake assays were performed either in the presence or absence of three different following compounds: vemurafenib, a chemotherapeutic drug used to treat melanoma; cimetidine, an antacid or antihistamine, known inhibitor of cation transporter mediated uptake; or dasatinib, another chemotherapy drug used to treat leukemia. We can see that in samples treated with cimetidine and dasatinib, uptake of both levofloxacin and metformin were profoundly inhibited. In comparison, vemurafenib didn't have any effect on the uptake of either of these two drugs. This provides some insight to drug developers, suggesting that prescribing levofloxacin or metformin to patients taking dasatinib would not be recommended. However, they would be safe to prescribe patients taking vemurafenib. So if you want to perform a similar drug-drug interaction study to monitor the uptake of your compounds of interest, these SLC overexpressing RPTech cells could be great tools for your study. In addition to testing renal transport and drug-drug interactions, SLC transporter expressing RPTech can be used to measure nephrotoxicity. In this study performed by our collaborators at Merck, cell viability assays were used to test the toxicity of some analogs of antiviral drugs. The three compounds that were used in this study were tenofovir disoproxil fumarate or tenofovir DF, tenofovir and cidofovir. We have tenofovir DF on the left, tenofovir at the center, and cidofovir on the right. Parental HTAT immortalized RPTech are indicated by the blue curves, while the OAT1 expressing RPTech are in yellow. You can see that the ATP concentration curves on both RPTech cells follow a downward trend for tenofovir DF. However, the curves diverge for tenofovir and cidofovir. The parental cells were unaffected by the OAT1 expressing cells showed. Oh God, I messed up once again. Brian, sorry. I need to start.
It's okay. It's okay. Do you need to get up and grab a drink or anything?
I'm fine.
Okay.
Okay, slide 16 one more time. In addition to testing renal transport and drug-drug interaction, SLC transporter expressing RPTech can be used to measure nephrotoxicity. In this study performed by our collaborators at Merck, cell viability assays were used to test the toxicity of some analogs of antiviral drugs. The three compounds that were used in this study were tenofovir disoproxyl fumarate or tenofovir DF, tenofovir, and cidofovir. We have tenofovir DF on the left, tenofovir at the center, and cidofovir on the right. Parental H-dot immortalized RPTEC cells are indicated by the blue curves, while OAT1 expressing RPTECs are in yellow. You can see that the ATP concentration curve of both RPTEC cells follow a downward trend for tenofovir DF. However, the curves diverge for tenofovir and cidofovir. The parental cells were unaffected, while the OAT1 expressing cells showed a higher toxic effect with these two drugs. These demonstrated that renal toxicity is OAT1 dependent for tenofovir and cidofovir, but not for tenofovir DF. So overall, we have several pieces of data indicating that H-dot immortalized primary RPTEC that expressed SLC transporters have multiple applications for toxicity testing. In summary, ATCC's primary and H-dot RPTEC display many key characteristics of renal proximal tubular cells. You can get the solute carrier protein overexpressing H-dot immortalized RPTEC cells, in which uptake of the corresponding anionic or cationic substrates are enhanced compared to the parental cells. And OAT1 or OAT3 and OCT2 inhibitors can also inhibit the uptake of corresponding substrate. Finally, some of these SLC overexpressing cells can very well be used for drug-drug interaction studies, as well as they have nephrotoxicity applications. Let us now talk about ATCC's offerings in cardiovascular models and some of the latest progress that we have made with immortalized cardiovascular cells. With cardiovascular diseases causing 1/3 of deaths worldwide, there is an urgent need to develop therapies to treat and cure this threat to global health. Advances towards these cures depend upon an improved understanding of the etiology and molecular mechanisms underlying these diseases. To achieve this goal, the scientific community is engaged in active research on topics such as basic cardiovascular development and angiogenesis, metabolism, regenerative medicine including tissue bioengineering, cardiovascular and blood diseases, cardiovascular effects of inflammation, vascular and lymphatic disorders, hypertension, and atherosclerosis. As can be seen in the figure on the top right, endothelial cells form the lining to the inner lumen of vascular tissue and are central to many of the ongoing research themes. Let's now take a few moments to discuss one particular cardiovascular research topic, angiogenesis. Angiogenesis or blood vessel formation occurs when new capillaries sprout from pre-existing blood vessels or capillaries. This process plays a central role in tissue development, wound repair, reproduction, and several human diseases such as cancer and age-related macular degeneration. As a cancerous tumor grows, it requires an increasing supply of nutrients and oxygen. To obtain this via the body's existing circulatory system, the tumor sends signals that stimulate new blood vessel formation that extends the blood supply directly into the tumor. To impede this process, numerous anti-angiogenesis drugs have been developed, including several that have received FDA approval. Researchers use in vitro models of angiogenesis to gain a better understanding of the signaling pathway that influence angiogenesis under normal or hypoxic situations and screen for activator or inhibitory compounds. ATCC has several immortalized primary endothelial cells that can be used as models during cardiovascular research. Our most popular model is the immortalized human umbilical vascular endothelial cells, or the HUVECs. We also offer TIME, or telomerase immortalized endothelial cells, which is derived from the dermal microvascular endothelial, and the telohec that was immortalized from a human aortic endothelial cells. In addition, GFP tagged and NF-kappa B reporter derivatives of both cell lines are also available. As expected, they all display expression of endothelial marker CD31 and take up acetylated low-density lipoprotein, which is the hallmark of endothelial cell function. And finally, earlier this year, we launched our newest cell model, HPAC-BMI1, which are immortalized human pulmonary arterial endothelial cells. These cells were generated by the stable overexpression of the BMI1 protein. In the next few slides, I will take some time to introduce this new offering. Pulmonary hypertension is a medical condition which affects 1% of the global population, with over 100,000 newly diagnosed patients in the US each year. It occurs when the pulmonary arteries narrow and stiffen, that causes increased blood pressure inside the lungs. Human pulmonary arterial endothelial cells are a key cell in the etiology of hypertension, and ATCC's immortalized HPAC BMI1 can be a valuable tool to gain a better understanding of the condition and develop therapies to treat and cure this disease. So as mentioned before, ATCC's latest offer in cardiovascular cellular model is a BMI1 immortalized HPAEC line. The cells display morphology and physiological characteristics similar to the primary HPAECs. As shown in the bottom left graph of the immortalized HPACCs, can grow 35 population doubling and beyond without any change in cellular physiology. They form capillary-like tubes and readily uptake acetylated LDL, both of which are key characteristics of primary HPAECs. The micrographs in the bottom panel of this slide shows HPAEC BMI1 cells incubated with Alexaflor 488, acetylated LDL, and its uptake was monitored by a fluorescence microscope. We can also see the formation of capillary-like structure by the immortalized HPAEC. Here, the capillary structure can also be disrupted by sunitinib, an angiogenesis inhibitor. The images here are from an endothelial cell tube formation assay in 3D culture on gel matrix, where capillary-like tube formation by HPAEC BMI1 cells and inhibitory effect of sunitinib is monitored under a phase contrast microscope. Let us talk about how ATCC's endothelial cells can also be used in co-culture models, where two or more cell types are cultured together. Co-culture models have been developed to better represent the in vivo environment. For example, culturing endothelial cells in a cell culture plate alongside fibroblast allows the formation of more heterogeneous tubules without the requirement of ECM coating, which often is not suitable for high-throughput studies. In response to this interest in more physiologically relevant models, scientists at ATCC optimized a co-culture system using ASC5 our HTART immortalized mesenchymal stem cells, and the GFP-expressing aortic endothelial cells, telohec. This system forms tubular structure in under 7 days compared to 14 days seen in standard co-culture method. Immunofluorescence at day 7 shows that the MSCs co-cultured with the microvascular structure have differentiated into smooth muscle as demonstrated by the alpha smooth muscle actin staining. This method is also compatible with multi-well plate formats, making it amenable for use in high-throughput assays such as compound library screening for activators or inhibitors of angiogenesis, as we shall discuss in the upcoming slides. We studied effect of various angiogenesis promoters and inhibitors on this co-culture model and found stimulators like VEGF promotes angiogenesis. In the upper panels of this slide, we see a dose-dependent increase in the tubule formation as observed through the GFP-labeled telohex. On the other hand, angiogenesis inhibitors can disrupt the tubular structure as depicted in the 3 graphs at the bottom panel. We have used 3 different angiogenesis inhibitors, such as suramin, which blocks various growth factors such as IGF, EGF, sunitinib, which is a receptor tyrosine kinase inhibitor, and bevacizumab, or Avastin, which is a well-known chemotherapeutic drug that blocks VEGF. As seen in the bottom panel, all of these compounds affecting different pathways disrupted the tubular growth. The last story that we would like to talk about is how ATCC's co-culture model using ATCC's H-dot endothelial and mesenchymal cell lines helped in screening tumor angiogenesis inhibitors. This work was done in collaboration between ATCC and Dr. Menghang Xia's lab from the NCATS. In order to understand the premise of the study, let us go through the mechanism of hypoxia-induced angiogenesis. The core of a tumor can be oxygen deprived, which can result to a hypoxia-induced angiogenesis. Hypoxia-inducible factor 1 or HIF1 is a key transcription factor that regulates the expression of genes responsible for the survival and adaptation of cells as they move from normoxia to hypoxia. In normoxic condition, HIF1 undergoes proteasomal degradation. However, under extreme hypoxic condition within tumor, non-malignant cells or tumor-associated endothelial cells, HIF1 is stabilized and binds to the VEGF promoter, mediating VEGF upregulation. Additionally, EGFR signaling acting through the SRC and MAP kinase cascade induce STAT3, which also helps promote VEGF transcription. Hypoxic tumor shown in purple in this cartoon releases VEGF that establishes a gradient from the tumor to the nearby blood vessels. VEGF stimulates release of matrix metalloproteases and plasmin that help mediate digestion of extracellular matrix proteins, facilitating migration of both endothelial cells and tumor cells. In response to the VEGF gradient, some endothelial cells acquire a tip cell phenotype, pushing long extension towards the tumor. These tip cells will eventually form the new blood vessels and will provide blood supply to the growing tumor. Going back to our collaborative paper, Dr. Xia's lab used our angiogenesis co-culture model to screen compounds which can be used as potent angiogenesis inhibitor by inhibiting the HIF1 signaling and eventually serving as chemotherapeutic drugs suppressing tumor growth. As an example of the assay they ran using ATCC's angiogenesis co-culture system, they tested the anti-angiogenic properties of several identified HIF1 inhibitors. After 3-day exposure, all of the test compounds were able to block angiogenesis. In the fluorescence image of GFP expressing aortic endothelial cells, we can see a clear inhibitory effect of mycophenolate motafil on angiogenesis. The graphs in the lower panel are tube formation and viability of the angiogenesis co-culture system after 3-day exposure to the PI103, tranitinib, mycophenolate motafil, and niclosamide. All of these compounds tested were able to completely block angiogenesis. In another study, Dr. Shia and her co-workers set out to identify angiogenesis inhibitors using ATCC's GFP-labeled angio co-culture model in a high-content and high-throughput screening platform. They evaluated 2,816 compounds from the NCAT's pharmaceutical collection. Many of these compounds were chemotherapeutic agents already in the market, and others were in the pipeline. Following the treatment of the angio co-culture system with the compound library in 1536 plate format, they incubated the cells for 48 hours and measured the fluorescence intensities to obtain the dose response curves. In this paper, Shia and her colleagues identified over 100 compounds with anti-angiogenic properties. The left panel in the slide is the dose response curve plotted. Sunitinib, shown with the dark red dots, were used as positive control. The green dots represent strong angiogenic inhibitors, the blue dots weaker inhibitors, and the compounds denoted with gray dots did not have any anti-angiogenic property. Interestingly, in addition to identifying well-known anti-angiogenic and anti-cancer compounds, the study also identified quite a few novel compounds. Among all the identified anti-angiogenic inhibitors, the study found 19 compounds involved in the HIF1 and NF-kappa B pathways that we discussed before. The heat map on the right panel gives us a simultaneous visualization of the compound's potency and efficacy in angiogenesis and HIF1 and NF-kappa B pathways. Together, from these collaborative studies, we found ATCC's angiogenesis culture system to be an excellent tool that can be used in drug discoveries to target angiogenesis. In summary, ATCC immortalized primary cells alone or in combination are a user-friendly solution for building reliable and predictive cardiovascular cell models. Co-culture of immortalized mesenchymal and aortic epithelial cells provide a physiologically relevant platform to examine angiogenesis. Various angiogenesis promoters and inhibitors can be tested using this system in both low and high throughput format. To conclude, ATCC offers a variety of authenticated immortalized cell models. These cells save time and cost of the researchers in trying to immortalize these difficult cell types on one's own. Our immortalized primary cell offer primary cell functionality along with continuous cell line longevity. We have improved supply and reproducibility for a standardized model. And immortalization also eliminates donor variability. Immortalized primary cells, alone or in combination with other cells, are a user-friendly solution for nephrotoxicity or cardiovascular studies. Finally, to get the complete resource information on ATCC's immortalized cell portfolio, please visit www.atcc.org/hdart.
Well, thank you, Sujoy and Jonathan. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The session will be archived on the ATCC website, www.atcc.org. Let's go ahead and jump into our first question. Jonathan, do the immortalized kilohex cells still retain the characteristics of endothelial cells?
Hi, Brian. Yeah, great question. We've investigated the important features of endothelial cells, including the continued expression of CD31 and the uptake of the acylated LDL, and shown that these cells continue to maintain those features and functions. In addition to that, we've looked at the inflammatory response of these cells in response to TNF alpha treatment, and we've seen the inflammatory responses with the upregulation of the CD54, CD62E, and CD106 surface proteins. And these look all equivalent to what we would anticipate and have shown looking at the primary cell equivalent. So extensively taking a look at the functionality of these cells and shown they maintain all the function that we would expect to see from the primary cell equivalent.
Nice, nice. Now, can you talk a bit, Jonathan, about the parameters that we use to confirm that the cells have become completely immortalized?
Yes, that's a really important step, and there's a lot of work that goes on during the development phase of these cell lines to ensure that we can confidently call them truly immortalized cells. There are several things we look at. We look at proliferative capacity of the cells beyond the limits that can be achieved with the primary cells, and we will keep them in culture for over 35 population doublings to show that they are immortalized and can maintain that extended proliferative capacity. Additionally, we'll take a look at the telomerase expression using the TRAP assay to show that the telomerase has been upregulated and is maintaining function, which is enabling that immortalization. And then we'll also take a look at the karyotype and the phenotype markers of these immortalized cells once they've demonstrated extended proliferation to confirm they maintain a normal or near normal karyotype and the hallmark functions of the primary cells they were derived from.
Great, great. Now, the audience member in this next question wants to know how they can quantify the efficiency of angiogenesis.
That's a wonderful question. So for quantifying the efficiency of angiogenesis, most researchers take a look at the length of the endothelial cell tubules formed over a square millimeter or some other area designated. And we've utilized imaging software such as the free-to-use ImageJ software or similar software platforms that can take a look at the image and quantify the amount of tubules that have been formed over a certain area. There are automated image acquisition analysis platforms available in the market, especially for those people looking to do high content screening in multiple conditions and multiple wells. Those can be a very powerful tool to allow them to look at a large library of compounds and get standardized, reproducible data readout.
Okay. Now, I know that you addressed this in the talk, but could you talk a little bit about the difference between primary vascular endothelial cells and immortalized cell lines?
I'd be happy to do that. So primary vascular endothelial cells are derived from primary tissue, and because of the well-known Hayflick limit, have a population doubling of no more than about 15 cycles, at which point they'll kind of become senescent and eventually die off in the culture. In contrast, our immortalized vascular endothelial cells are able to go over 35 population doublings without any change in the functionality of the cells indicated by the expression of the CD31 marker and the uptake of the acylated LDL.
Okay. So a related question actually just kind of came in from the audience. Do you think that there's a drift in the function the longer that the cells are in culture? How might one address this? Or is there just a ceiling of how long you want to go?
Yeah, that's a great question. So as part of our development work, we routinely ensure that the cells can be maintained, so 35 population doublings, and maintain functionality. We haven't tested far beyond that. We don't anticipate any significant change because so long as the cells are maintained under correct culture conditions with the inclusion of the antibiotic selection to ensure there's a selection pressure to maintain the plasmid and expression of the telomerase enzyme, we believe that there should be maintenance of the extended proliferation and the rebuilding of telomeres. However, as part of our development, we chose a cutoff point of 35 population doublings. We would recommend coming back to the source after you've done 35 population doublings and start with a fresh culture, because then you have the confidence and the guarantee that they will maintain all the hallmarks of the function that you anticipate for the period of time that we have tested out rigorously. Beyond that, we believe they should be functional, but we don't have data directly support that claim.
Very nice, very nice. All right, so could you talk a bit about the media used to culture the RP-Tech SLC transporter cells?
Yeah, that's a great question, and we always endeavor to maintain that the culture conditions that we put our immortalized primary cells or any derivative should be equivalent to the parental lines they come from. We want to make sure you can maintain the same culture conditions that have been optimized for the primary cells and maintains the utilization of the immortalized cells to ensure that there's no phenotypic or functional changes due to the culture environment. So for our SLC transporter cells derived from our immortalized RP-Tech line, they use the same media system from the RP-Tech immortalized cells they were derived from, which is the DMEM-F12 basal media supplemented by our proprietary supplementation available under the catalog number ACS 4007.
Good, good. So I like this next question a lot. How many copies of the transporter gene were inserted into, I would imagine this would be the RP-Tech cell?
Yeah, it's a great question, and really excited to get under the hood and the work done to develop this line. And copy number variants and insertions is really important to think about the quality of cells when you haven't created too much genome instability. So as part of the development of these lines, we took a look at the copy number insertion using digital dPCR, and we showed that there are 5 copies of the OAT1 inserted into the OAT1 line, and 3 copies of both the OCT2 and the OCT2 inserted into the RP-Tech OCT2 line. So different experimental runs, different clones, but with either 3 or 5 copies were inserted into the parental line to express the OCT2 or OAT1 transporter, respectively.
Okay. Let's see what other questions have come in. Righty. Should you use antibiotics in the culture medium for the HPAEC BMI1 cells? And if so, can you talk about the concentrations?
Yes, so as part of the maintaining the selection pressure on the cells to ensure the cells continue carrying the the telomerase, which will ensure the functional immortalization of the cells, we do recommend the addition of selection conditions. In this case, we recommend the addition of lactocidin as a selection marker, and we recommend adding 4 micrograms per mil to your culture media to ensure that the sufficient selection pressure is maintained, that the line continues to express the telomerase enzyme.
All right, good, good. Let's see. We've had quite a few other questions come in. Alrighty. So the next one is not related to one of the cardiovascular or renal cells, but actually to the immortalized melanocyte. Do the H-term immortalized melanocytes develop pigmentation, meaning do they secrete melanin?
Yeah, that's a great question, and this is a really exciting model that we have developed for use in modeling the dermal tissue. We paired with some of the other immortalized dermal lines we have at fibroblasts and keratinocytes. And yes, these immortalized melanocytes do generate pigmentation, and when you put them into air-liquid interface 3-dimensional structures, you can see the deposition of pigment in those structures. If you want to learn more about this line and some of the fantastic work that the team has done developing three-dimensional models, there actually is an on-demand webinar available on the ATCC website. This was aired a year ago at this point, and the title, if you're looking for it, is 3D Models for Absorption and Cosmetic Toxicity Studies, where we focus on our primary and immortalized epidermal cells. It's great webinar and a lot of detail about the experimental work done to develop and characterize those lines and utilize them in those three-dimensional models.
Great, great. Now, donor variability assessments is critical for understanding how different patients can react to therapies, different mechanisms. Do we have different models from different donors?
That's a great question, and yes, it's really important. It's one of the values and the advantages of the use of primary cell material is that you can get material from multiple different donors. It allows you to capture that natural human variability. We generally use one donor to immortalize and create a clone of. So you have a standard reproducible model. For some of our models, we do have multiple donors. For instance, with our melanocytes, we have an immortalized adult and neonatal melanocyte line that can be used to get some of that variability. And it's an area we continue to think about and work on in our development teams to add more donor variability with additional donor material of immortalizing cell lines that we already have in the collection, to be able to provide a collection of lines which capture some of the naturally occurring variability in the population.
All right, great. Well, at this time, we will conclude our Q&A session. I'd like to thank Jonathan and Sujoy for the excellent presentation, and thank you everyone for attending this webinar. If you'd like to learn more about ATCC toxicology testing tools, please navigate to www.atcc.org/tox. And if you're attending SOT 63rd Annual Meeting and Tox Expo this year, you can talk to an ATCC expert at booth 401. Finally, we have an excellent webinar presentation on H-immortalized pre-adipocytes that you can watch on demand at www.atcc.org/webinars. So thank you again, everyone, for attending, and have a great day.